E. coli bacteria in the gut of mammals metabolizes lactose, a sugar commonly found in mammal diets. Lactose does not easily diffuse across the E. coli membrane, so permease actively transports it across. Once inside, lactose must be broken into glucose and galactose to be used, a reaction that is catalyzed by beta-galactosidase. The lac operon is an example of a negative inducible operon, meaning a repressor is always attached and mostly prevents transcription until another molecule, allolactose, binds to the repressor and releases it from the operon to allow RNA polymerase to bind to the promoter and transcribe. LAC-Z encodes for beta-galactosidase, LAC-Y encodes for permease, and LAC-A encodes for transacetylase. RNA polymerase can bind to the promoter and transcribe alone, but is not very efficient. Catabolite activator protein, or CAP, binds upstream of the promoter region and helps RNA polymerase bind more efficiently to the promoter. In the presence of both lactose and glucose, allolactose is produced so the repressor is non-active and the operator is clear. However, very little CAMP is made, so there is limited binding of RNA polymerase and there is small amounts of transcription. In the presence of just glucose, allolactose is not produced. Therefore, the repressor is binded to the operator and RNA polymerase is unable to transcribe. In the presence of just lactose, lots of CAP are produced. In addition, allolactose is produced, removing the repressor from the operator and allowing RNA polymerase to very efficiently transcribe DNA. In the presence of neither glucose and lactose, nothing happens. Thus far, we've been talking about wild type. Now let's talk about mutant type. Here I have mutations on the promoter and the operator. The genotypes for the lac operon are LAC-IS, which is the superrepressor, meaning the repressor cannot be inactivated by the inducer. LAC-OC, which is a constitutive operator, meaning the repressor cannot bind. And LAC either minus or plus, minus being unable to bind or be produced, and plus meaning a normal gene. When the promoter is mutated, RNA polymerase is unable to bind, and transcription does not take place. But since this strain is diploid and has a functioning copy of LAC-P, transcription will continue. The LAC-OC mutation is dominant over the LAC-O positive and will prevent the repressor from binding to the operator site, thus allowing RNA polymerase to transcribe the structural genes whether allolactose is present or not. Meaning, in the presence of lactose, or not in the absence of lactose, and just glucose, the operon will produce beta-galactose and permease.